Welcome to Sam Jill's Bioscience. This is the channel which is going to provide you about the concepts of complex biological topics and also going to address some of the key questions in a solution-oriented way. So today's topic is about the cellular confluency and cell splitting in a laboratory, and our focus would be especially with the animal cell culture. So first of all, I'd like to tell you about the confluency and how this when the cells grow, what are the phases that they go through. So in the left side of this slide, you see that there are different confluences. So when you see the cells, when you see under this microscope your cells, you see of the different, how would you see the different confluences? For example, like the left corner, if you see the very less number of cells, they are called 10%. So it's kind of an eye estimation that you have to be used with your observation, and you have to decide by your own. And it's rather empirical, like you derive it along the way you experience. So the slightly higher here, you see that this is the 30%, and then this is 50%. And where it is, you see that there are more number of cells and most of the space is occupied over here. So that is called that is 90%. And I'd like to also tell you about how the cells grow, or what are the phases the cells go through when they grow. So it's like the bacterial cells again. So they have this lag phase, which is this phase like when the cells actually prepare. When you seed the cells, just after the seeding, the cells are in a period of lag phase where they actually prepare themselves for the growth or proliferation. So then you see there is a steep rise of the cells. And here the y-axis tells you about the number of the cells, and this is the time. And so you can see that there is a log phase cells, and afterwards there is a stationary phase. That means the cells do not proliferate any further and they stop. And if you don't change the media in this, then the cells would not any further would be viable, and they would start ceasing or they start dying, and that is called the death phase. So you have to be always cautious when you're dealing with the cell lines that the cells are in the log phase, or which is also known as the exponential phase, where they grow exponentially, and that's why you see this steep rise. And this is the reason the cells need to be splitted. That is called subculturing, to maintain the genetic stability, the phenotypic status, and they remain viable. So I just shortly tell you about like how what are the stages of cell culture. Although my next videos that you're going to see, you'd see in detail about all the methods and all, how you do it and why you do it, and how would you troubleshoot what kind of problems that arise, and how would you going to troubleshoot. So usually this is a flow chart that's in a very simplified way I'm telling you, but please click on to my other videos, then you can have a look that I have explained it each step of this flowchart in a very detailed way. So the first thing is the seeding of the cells, and then the cells grow. You put some media, and here every 42 to 78 hours, 72 hours, you have to change with media. But it is like it varies from one cell line to other cell line, or how you split the cells. But usually this is the norm, that every alternate day you should you should change the medium, because you have to, otherwise if you do not replenish with the new medium, then the cells would die. And once they reach around 70 to 90% confluency, then you trypsinize the cells. And once you trypsinization means that you take the cells, because you're working with the adherent cells, so you just trypsinize the cells and you subculture them with the appropriate dilution factor. So I just tell you about a short thing about this. Is a very common question that often appears when you are doing in the lab, or you can say this is a routine thing that in a laboratory you do. That is split cells from one confluencer to another confluencer, because you might need the cells just to simply to subculture, you might need the cells for one experiment, so it might have different purposes. So here I'm going to tell you about the calculation, that how you do it. So for example, the question is, you have cells and you want to split cells from 80% confluencer to 40%. Well, so now it is a very vague question. To be very specific about this, what I'm telling is that suppose you have a T75 flask. 75 is this is the surface area of the cells. That's why we call T75. And you have cells, you have already seeded the cells, and you have 80% confluency. And now you want to split the cells. You want to split and seed the cells again in another flask, which is also T75. But you want the cells to be around 40% confluence. So how would you do that? So this is the question. So this is a very simple calculation. You just need to understand this is simple mathematics, simple calculation. So as I said that this is 75. So this is 75 means 75 centimeter square surface area, and 80% confluence means you have 80% of the 75 centimeter square area occupied. That means 60 centimeter square area is occupied by the cells. So this is what you have. And now what you want to have. So you need to have 40% confluence. So what would be the area in terms of 40% confluence if you just go through the in terms of your surface area? So that means 40% of your 75 centimeter square area. And when you calculate, it is 30. So that means you have 60 and now you want it to in 30. So what you do is that usually I already described you about the steps, like you have the cells and then you want to you trypsinize the cells, you centrifuge, and then you have the pellet. So you have all the cells in one vial or one falcon tube, and then you remove the old media and you add 1 ml of fresh media. So now you have all of your cells that were in your entire flask in one vial, and that is 60, like this area occupying things, this area occupying cells are in 1 ml. And now you want to seed the cells in a way so that it is 40% confidence to the T7, another T75 flask. So it's a very simple calculation. So you just calculate that you want to achieve this much of confluence because you have this much of cells, this much of cells are in 1 ml. Now you calculate it half and you want the half. So you need half of it. So that means you need 500 microliter of it. So now I give another example to make it clear. So the earlier one was easier. So this is also the same way you calculate. So now you have the T75, 80% confluence. The only difference is you want to set it into T25. That is T25 means the surface area is 25 centimeters square. So you do the calculations in the very same way. That is, you have what you have, that is 60 centimeters square is occupied, whereas you want 10 centimeters to be occupied. So you do the same thing. You pull down your cells, you centrifuge and you get the cells in one vial. You remove the old media, you add 1 ml of your media, of fresh media. So now you all the cells that were occupying your 60 centimeters square in your one falcon. And now what you do is that you just want to achieve, well, I want to take those cells which would be occupying 10 centimeters square in this T25. So you calculate in the same way and you get 166.66. You can make it a round figure and then you could see the cells in such a way. So this is a very simple way of calculation, and I have next videos coming on other animal cell culture details and about other biological things. Some of them are very bench oriented and some of them will be very concept oriented, text oriented. So if you have liked my video, please subscribe, and I'm happy to help you always to solve with your questions. Thank you very much.